Today we will be discussing stem cells. The first thing we need to do is define them. So what exactly is a stem cell? Stem cells are single cells that can replace themselves with the ability to differentiate into other cells. Or in other words, undifferentiated or primordial cells of an organism that are capable of producing more cells of the same type from which other cells can be produced by differentiation. You may be asking yourselves, where do these amazing things come from? Well, they come from any multicellular organism, like plants or animals, but we will be covering mammalian stem cells. In mammals, there are two types: embryonic stem cells, which are what we will be focusing on, and adult stem cells, which are also called somatic stem cells. Embryonic stem cells come from blastocysts, while adult cells come from tissues. Both of these are formed and harvested in similar ways. Embryonic stem cells divide through mitosis in order to produce more. They are harvested from the blastocyst and propagated in petri dishes, or something similar, to develop pluripotent embryonic stem cells. The main reason as to why these cells are so important is because of its range of uses. We could spend a whole video on this topic alone, so we will just go over some of them. They can be used in cancer therapy, the creation of test tube babies, and regeneration. Regeneration in particular is fascinating simply because of the possibilities it presents. The mechanics of stem cells allow for regeneration, letting the process of renewal and restoration continue. This is hopeful for those affected by tissue and organ damage, even going as far as lost limbs. As mentioned before, there are numerous benefits provided by stem cells. They can help save lives and even create them. Regenerative medicine adds to the former, and the concept of test tube babies no longer seems like a crazy theory when related to the latter. It can help us explore new areas of biology and medicine, even the testing of new drugs and cell-based therapy. The potential to replace neurons, fixing damage caused by Alzheimer's disease or Parkinson's disease, producing insulin that can aid in the growing problem of diabetes, or repair the destruction of muscle is great. Like anything else, people have objections to the use of embryonic stem cells. The harvesting of stem cells means destroying a human embryo, which people oppose because it involves the killing of a human before they are even born. There is also an issue with the distinction between creating embryos for research and reproductive purpose. The creation of new life unnaturally is the line that many aren't willing to cross. Along the same path is the problem of cloning. Again, it isn't exactly natural, and the ethics against giving a life to a living organism that isn't theirs is something people have morals against. And then there is the deep topic of human/non-human chimeras. Luckily, there are new alternatives that are available to embryonic stem cells. Skin cell reprogramming is something that is currently getting more study on. Research on adult mouse skin cells have proved that it can yield cells that looked and acted like embryonic stem cells. However, it has yet to release definite answers. Amniotic stem cells are stem cells from amniotic fluid that can be harvested and harnessed, but they lack the same potential that embryonic stem cells, and very few can come from this. Cord blood stem cells come from the blood from the umbilical cord and can be taken and preserved for use. Unfortunately, the number of cells available is very low, but this is a great alternative. All across the world, there are various limitations placed on the use of stem cells. In the US, there are many laws passed to prohibit the destruction or creation of human embryos for medical research. Arkansas, Indiana, Michigan, North Dakota, South Dakota, and Virginia all ban human reproductive and therapeutic cloning. Iowa, Maryland both ban human reproductive cloning. For now, though, we will just have to wait and find out the future of stem cells.